Hi again, my name is Yigit. I'm from İzmir High School from Turkey, and today I'm going to talk to you about my project, the dosage-dependent protective effect of melatonin and sulforophane administration on Alzheimer's disease model. So our introduction is that the average life expectancy is prolonged due to the increased quality of life and healthcare success. And Alzheimer's disease is characterized by the progressive loss of significant cognitive functions, especially those related to the memory. During the degeneration process, patients' brains undergo a number of morphological and functional changes, including synaptic loss, impaired neurotransmission, and decreased metabolism. And here we can see the process by which the amyloid beta proteins form the amyloid plaques in the interstellar space. And here in this image, this is how the APP forms, while the normal psychological pathway is visible in the blue marked part on the left here. The pathway in which the amyloid beta proteins observed in Alzheimer's disease are formed on the right. On the left, proteins formed by alpha secretase are soluble in water and do not precipitate. However, in the pink marked pathway on the right, amyloid beta formed by beta and gamma secretase is not easily soluble in water and tends to accumulate. And here we see the amyloid beta proteins accumulating around the neurons and wrapping neuron extensions in the Alzheimer's disease. So why did we pick sulforophane? Alzheimer's disease is not a result of normal aging. 40% of dementias can be prevented before they occur, and nutrition and sleep are among the most important steps to prevent this disease. So as a diet, first there's the Mediterranean diet, second the DASH hypertension diet. It's a type of diet applied to help treat high blood pressure. And then we have MIND diet. It's basically Mediterranean plus DASH diet. Is a combination of Mediterranean and DASH dietary habits and consists of minimally processed foods, including grains, legumes, vegetables, fruits, nuts, and fish. And sulforophane is a potent dietary bioactive agent obtained from the surface vegetables, has been extensively studied for its effects in the prevention and treatment of diseases. And one of these diseases is Alzheimer's disease. And why melatonin? Melatonin is a hormone found naturally in the human body that regulates the sleep-wake cycle. And it's also known to suppress the programmed cell death in the mechanism of the cells. And in this project, we aim to evaluate the dose-related effects of melatonin administration against amyloid beta toxicity. We had three hypotheses in mind. The first one being sulforophane administration protects neuronal cells against amyloid beta toxicity. Second one, melatonin administration protects neuronal cells against amyloid beta toxicity. And the third one was the co-administration of sulforophane and melatonin increases their protective effects compared to their separate administration. The materials that we used while conducting the project, we used the amyloid beta 2535. Then we use the CHCY5Y cell line, which is the human neuroblastoma cell line. Then we use sulforophane, and then we use melatonin. Then we have our findings, and here you can see each group's picture under the microscope. Then this is our first experiment's MTT results. And then we did another test to see the results again. So here are the second experience MTT results. And in conclusion, due to the increase in elderly population in the world, the incidence of the disease, the Alzheimer's disease, has increased and attention has been drawn to the development of an effective treatment against Alzheimer's disease. In our project, we successfully created the amyloid beta toxicity model, which is accepted as an in vitro model of Alzheimer's disease by applying AB2535 peptide to neuronal CHCY5Y cells. While planning our project, our main research questions were, does the presence of melatonin protect neurons from cell death against amyloid beta toxicity? And does the presence of sulforophane increase their protective effect? According to our literature review, we have found three hypotheses. First one was sulforophane administration protects neuronal cells against amyloid beta toxicity. Second one was melatonin administration protects neuronal cells against amyloid beta toxicity. And the third one, we suggested that the quiet administration of sulforophane and melatonin increase their protective effects compared to their separate application. According to the results of this project study, we showed that our first hypothesis is valid and that a low dose of sulforophane previously applied to the cells protects the cells against amyloid beta toxicity. In our second hypothesis, we have suggested that the administration of melatonin protects neuronal cells against amyloid beta toxicity. In our study, we showed that 1 micromolar dose of melatonin application had a protective effect against amyloid beta toxicity. In our third hypothesis, we suggested that the quiet administration of sulforophane and melatonin increased their protective effects compared to their separate administration. And in conclusion, in our study, it was evaluated that a high dose of sulforophane reduces cell viability against amyloid beta toxicity, while a low dose protects cells against amyloid beta toxicity. And the suggestions that we have for future studies is melatonin and sulforophane were used alone in the study, and their different doses and long-term effects should be investigated. The effect of sulforophane and melatonin were investigated in a single cell type. Other cell lines or human cells should also be tested. In the future, the effect of melatonin and sulforophane should be tested in animal studies in which Alzheimer's model is created. And thank you for Professor Dr. Punarakan and her lab members for helping me with my project and providing me her lab. And here are the sources that we use while conducting the project. And thank you for listening to me.
Yigit, that was wonderful.
Thank you.
And just let me know foremost, are you a ninth grader?
Yes, I am a ninth
grader. Are you
grader.
sure?
Yeah.
Is that confirmed?
Yes, yes.
Because the terminologies that you've used were not at all associated to a ninth grader, you get. So foremost, you have to show me your ID card so that I know you're a ninth grader, right? Okay. That was a little confusing. All right, Yigit, by the way, now I have a serious question to you. Why did you choose to work on Alzheimer's? I mean, this is a serious thing. No, people don't think about that mostly. So why Alzheimer's?
Last year, I had a project on brain cancer cells, and I used melatonin, and it was the first time that it was tested on brain cancer cells, the effect of melatonin, and I showed that the higher dose the melatonin you apply, the brain cancer cells die, the cell viability decreases. And that's why I'm interested in the brain. So I wanted to conduct another experiment this year again on a disease on the brain. And I picked the most common disease, which is after cancer on the brain. So I picked Alzheimer's for that reason.
But why brain studies, Yigit? I don't want to know the point of source and the point and the source of inspiration. People, you don't talk about studies, especially these chronic disease. So is there something that inspired you in your life?
I mean, because I picked Alzheimer's because it's a very common disease nowadays, because the age expectancy increases and there's a lot of elderly population in the world, and it's a very common type of disease. That's why I chose Alzheimer's.
And you, while you were completely talking about your research, you said we are researching. So who with you? I mean, were you researching in a team or a group, or were you doing it all?
I was alone in the study, but I said we because the professor provided me with her lab and helped me with the literature review. So that's why I said we. I didn't want to take any rights. That's why I said.
No, I just wanted to know if it's a group kind of a study that
no, it's just
goes
me.
on. It was you. And did you, because you said that you have already been winning a couple of competitions, national competitions with this research and all that stuff. So have people been contacting you for implementing it in somewhere? I mean, it's a hypothesis, it's in the hypothesis form, right? And the next stage of hypothesis is definitely it needs to be implemented somewhere so that we can get the real results out of it and have a success story, then know the impact and all that stuff. So what's the next step? Where are you heading with this research now? What are you planning?
I want to, because I'm a high school student, it's really hard to do animal studies because the ethical commission doesn't allow high school students to do experiments on animal studies. But if I get the chance, I want to do it on animal studies to show that it can, if it works on animal studies, I can show that it can also be conducted. It can also work on humans as well, and that's
what help do you need from people who can help you? Would you like to give a call over here now, because it's your time now? You people across globe are watching you. Who would you like to reach out and share your email address or the contact details, so that if people are watching you and would like to reach out to you to know further or to support you, they can?
If we can somehow get from in the future, if we can get acceptance from the ethical commission to accept our experiment to do it on animal studies, and we get the animals that we want, that's what I would like for the future. And should I share my email for this? How should I write?
Just say it, or go
okay.
to our live YouTube channel, and always, of course, write it in your in the end of the presentation whenever you make it the next time. Or probably go to the YouTube channel where you are watching live, where GIFT is being live, running live as a 24-hour hackathon. And write it over there as well. Say it loud over here, and definitely people are going to be contacting you.
Okay, so it's I will write it in the chat of the YouTube channel and say it here, and it's YIGITKAAN_KIZLIER@gmail.com. I also wrote it in the YouTube live.
Perfect, Yigit. And Yigit, along with you, we have a very young innovator with us. Hi, Philip. Philip was like, hello. Philip, I put you on spot. Unmute yourself.
Hello.
Hi, how are you, Philip?
I'm good.
We missed talking to you in the beginning, but I thought this is a good time we can talk to you. We have a couple of minutes. Philip, did you get to hear Yigit and his big terms?
Okay. Good luck for him.
Yigit, you hold here. I would like Philip, if he has some questions to ask you. He's the third co-host, right?
Okay.
So, yeah, Philip, did you get to hear Yigit?
Melatonin is also used for ADHD management. Would you consider exploring that area of research?
On ADHD? We did not have that in mind, and we only studied it. I only studied it on Alzheimer's and brain cancer. Maybe in the future, I can conduct a study on it as well, but it's not planned for right now. Thank you. By the way, I said my email wrong. Sorry for that mistake. It's not underscore, it's with a dot. I said it wrong, sorry.
Today is a wrong email address day. Don't worry, we have been having it since morning. So no worries at all. So we have a new day celebrating wrong email address day. That's cool. That sounds good. Yigit, I wish you, and we wish you, Team GIFT wishes you the best of luck for your future. I'm so impressed. And Yigit, it just skipped out of my mind while you were presenting it. Do you have any sort of idea that implicate, or maybe, for example, if even the 50% of what you presented in your hypothesis, if it turns to be, if it's like concluded as a fact, so how much it is going to be impacting on the overall reduction of Alzheimer's disease? Have you got those kind of estimations done?
No, but no, sorry.
All right. And any kind of estimation regarding the costing of whatever you said? The plan that you delivered. I don't want to take those big names at the moment, but any kind of cost?
The costing
per person.
The costing in our project wasn't that much, but the problem with it creating the cell line for the Alzheimer's disease was most of the part that we had to put money on. But other than that, melatonin and sulforophane are not very hard to find. So that was not the hard part. But as I said, creating the cell line was the hard part.
Perfect, perfect, perfect, perfect. Thank you, Yigit. Well done. And Team Gift wishes you the very best of luck for your future.
Thanks
Do come to us the next time and let us know where you are in your research till then and how many of your hypotheses have then been till then been concluded in a fact. Bye-bye.
Okay. Thank you. Goodbye.